Good morning to everybody. Good morning to the large family of MD Anderson. It's 10 a.m. It's Enjoy Science time. And I'm happy that you are here with us for the 6th week. We already have over 6,000 attendees. Thank you all of you, because without your time and your participation, certainly webinars make no sense. This is a webinar designed specially for clinicians, for scientists, for patients, patient advocates, in order to see what is the best at MD Anderson clinical and research fundamental science. It's done, Enjoy Science. It's hosted by myself and my friend and colleague Alessandra Ferraioli, under the office of Dr. Giulio Draeta. And of course, I have to thank Alison Gard, Liz Kentz, and Valerie Jimenez, because without their wonderful job, nothing can be done. It's attention to details to make these things happen. And with this said, I will push all of you to use the chat during the presentation for questions. There were wonderful questions during these presentations. And of course, remember, most of the presentation are already on the site of Enjoy Science, so you can go back and listen and see all this wonderful science. And it's my pleasure today to introduce you Yejing Ge. She is an assistant professor in cancer biology, but I think the definition is she is a rising star. She is a wonderful young scientist that we were very happy to have her at MD Anderson. She trained with Ellen Fuchs, and Raghu Kaluri and Siprit were able to steal her from other places and bringing her here under the sunny and warm Houston. Yejing is doing a type of science that very few are doing. She's checking on cell plasticity, which is basically an extraordinary phenotype which is the interface between normal and abnormal. She's checking on how the stem cells differentiate, how the cells accommodate their survival in different environments, epithelial mesenchymal transition, wound repair, and cancer. She has a wonderful list of publication, and what I remarked to her from the first moment I meet her quite 1 year and something ago, how's the time it's flying, Yejing, no? You just arrived at MD Anderson 18 months ago already. I remarked her wonderful attention to details, which basically it's a great sign of a careful and great scientist, because what makes science great are not only the ideas, but the attention to details. So today we have a speaker who is carefully thinking to science and carefully presenting the science, a young and wonderful speaker. So thank you for being with us, and Yejing, and good luck with your presentation. Thank you again.
Thanks, George, so much for the nice introduction. Can you hear me well? Okay?
Everything is fine.
Okay, great. All right, thanks, George, again, and thanks, George and Alexandra, for putting together such a fantastic Enjoying Science series and inviting me to here to speak. Such a privilege. As George mentioned, I started my lab at Department of Cancer Biology at the beginning of last year. I will take today's opportunity to share with you some of the work my lab is currently pursuing in the context of stem cell plasticity and skin diseases. I'm really new to webinar, and this is the first talk I'm giving in front of my computer. So please bear with me here. So in my lab, we're fascinated about these guys, adult stem cells, which are essential units for postnatal remodeling and wound repair. Now they come in different flavors. Some are constantly cycling, such as those in the classic hematopoietic system, the GI tract, the skin, and some are deeply quiescent unless confronted with injury, for example, the muscle or the brain. And there are other tissues where differentiated cells can be adapted to repair their damage, so-called the facultative stem cells, such as the lung, liver, or pancreas. These adult stem cells have remarkably adapted to meet the needs of each specific organ. Now, no matter how different they may appear, there's one aspect that is absolutely conserved. That is, they'll be able to respond to injury. Upon various insults, adult stem cells often expand their fates beyond their homeostatic patterns in order to cope with stress and mediate tissue repair. That's often known as the term plasticity. Now, plasticity is essential for organismal survival, but it also poses a vulnerability to individuals who undergo prolonged stress where stem cells could be subjected to functional exhaustion, for example, those observed in aging, or malignant transformation that occurs in cancer. Now, my lab uses skin as our model. We apply multiple techniques from mouse genetics, development biology, and functional genomic approaches. And we aim to understand the molecular mechanisms behind stem cell plasticity and how its deregulation leads to diseases, including wound repair, cancer, and aging. Now, mouse skin offers an excellent model to address questions in stem cell biology, and I hope to convince you today this point of why studying stem cell plasticity in vivo using mouse skin is advantageous. The mouse skin epithelium has many distinct lineages, just like human skin, and two of the main ones are hair follicle and epidermis, each harboring their own resident stem cells that are well defined. This is important as we could easily purify them with flow cytometry with well-defined surface markers. Now, one struggle in studying cancer is often we don't know what is a normal cell to compare to. And fortunately, we know that fairly well in the skin, and I'll come back to the point in a minute. These stem cells are highly accessible and abundant. This becomes highly relevant for us to be able to do genomic profiling in vivo as stem cell interface their unperturbed native niche, which, as I'll show you in a moment, is very different from looking at stem cells in culture in vitro. Now, we like skin also because they are genetically tractable. I will be talking about several genetic tools we use quite commonly in the lab. Finally, mouse and human skin biology share many commonalities, therefore using mouse models relevant in understanding human biology. Now, stem cells of the hair follicle reside at the base of the follicle, known as the bulge. And during normal homeostasis, they feel the cyclic bouts of hair follicle regeneration and hair growth. By contrast, epidermal stem cells reside in the innermost basal layer of the epidermis, where they generate an upward flux of differentiating cells that produce the skin barrier. There are key transcription factors defining each lineage. SOX9 is known as a master regulator of hair follicle stem cell identity. Now, SOX9 is very important for many organs. In the hair follicle, though, it is required for stem cells of the follicle to maintain their function. Now, in the epidermis, we've recently found that a KL5, which is a Krupp-like family member, is a crucial transcription factor. During development, as you can see from this immune fluorescence image, KL5 marks the epidermis and declines as SOX9 rises in the newly emerging hair follicle precursors. Now, upon injury, both hair follicle and epidermal stem cells near the wound site can mobilize toward it, drive the re-epithelialization of the wound bed, and restore the barrier. Lastly, both lineages can give rise to squamous cell carcinomas, which is quite common in the skin. And when they acquire oncogenic mutations such as RAS, P53, these common ones. Now, in our lab, we've recently set up this in utero lentiviral injection system where ultrasound-guided micro-injection platform delivers lentiviral particles directly into the amniotic cavity of living pregnant mice, such that her embryos are now bathing in a lentiviral soup. It is a 20-minute surgery, and we put the pregnant mom into sleep and inject lentivirus into her embryos. Now, what's neat about this technique is that by virtue of the fact that the lentivirus infects the first layer it encounters, and that the skin exhibits a single layer of surface progenitors at the time of embryonic development, which is E9.5, one can actually achieve skin-specific and stable transgene integration in an extremely high efficiency. What I will do is use a very quick slide of our previous work on microRNA screen as an example to illustrate how this technique can be utilized for functional screenings to uncover oncogenic drivers in vivo. So First of all, what we did was to engineer a microRNA expression vector to which we named small accurate SA mirrors. I will spare you the details of how we modify the backbones, but the idea here is we want to express microRNAs that are precisely processed at the 5' end, which is critical because that's where the seed is. Now we focus on microRNAs found to be overexpressed or associated in squamous cells, since these are potentially oncogenic in that context. And finally, in some cases, we might make the library constructs also for the star strand if these are highly expressed, so that they can be examined independently from the mature strand. So this is our library. Now here's how we perform the screen. We pulled the SA mirror lentivirus expression microRNA that are bounded in tumor. That makes our library just below 200, including spike in scramble controls, very sweet site to work with for such in vivo screens. So we infected the skin with our library using the in utero approach. We aimed at low multiplicity of infection so that each cell received no more than one vector, and we injected a sufficient number of embryos to achieve a reasonable coverage. Then we let the mouse born, and we apply the carcinogen DMBA TPA to the animals at P50, which typically selects for HRAS mutations. We collect the tumor in the end, isolate the genomic DNA, amplify the SA mirror inserts, and subject them to deep sequencing. So the idea here is by pushing the system into tumor genesis, a putative oncomere, when overexpressed, is expected to drive tumor formation, therefore being positively selected for in these tumors, which can be read out as enriched read frequencies of ACE mirror insert in a given tumor. So here's a snapshot of the top hits. Each pie represents read distributions of SA mirrors in a particular tumor. In parentheses is the number of tumors sharing the same major microRNA hit. A striking trend, as you can see here, is that the majority of tumors are dominated by one particular microRNA, suggesting the monoclonal nature of some of the tumors. And those selected microRNAs are our top candidates. For example, among 107 sequenced tumors, 22 of them are dominated by MIR21, a known oncomere in many other types of cancer. I'll come back to MIR21 in a moment. Notably, this clonal dominance is phenotypic only for the tumors and is not seen in the tumor adjacent skin. Therefore, it is not simply due to DMBA treatment. And also E12.5, which is the point where we just did the infection, or P50 before the tumor onset, we don't see that selection. So ruling out the unspecific artifact from the library itself. Long story short, among our top candidates, we then went on to show some of them exhibiting SCC promoting activity in functional assays in vivo. And interestingly, some of their high expression often correlated with more advanced tumor grade and stage, as well as poor patient survival. So together, I hope this study serves as a proof of principle where we could exploit such technique as a non-germline germ model to identify oncogenic drivers in vivo. Now, with these ground sets, for the rest of today's talk, I will be focusing on two stories, one about cancer and one about aging. So in both cases, I will be talking about our current ideas in the context of my previous work as postdoc fellow in Ellen Fuchs' lab. Now, please take my words for it that in the process of getting some really interesting preliminary data before the pandemic hits, we're hitting something really exciting, and I'll try to share a bit of those during the talk. But my main goal today is to get you all excited about skin as the lab does. And so our first story is about cancer as a non-healing wound. So as I just mentioned to you, KL5 and SOX9 mark the epidermal and hair follicle stem cells respectively. And each are distinct stem cell population in a normal adult skin. So it came to me as a big surprise that what we found that KL5 and SOX9 are actually co-expressed in squamous cells of the skin in cancers. Now, these are human skin SCC cells xenografted onto a nude mouse. You're looking at on the left the primary tumor and on the right the lung metastasis. Now, we refer to this phenotype as stem cell lineage infidelity, as it has been widely known in many other cancers actually. They really express mixed lineage markers, and that's why they are sometimes considered really bizarre. I actually think that cancer do not invent anything new. They always hijack. So many of you might have heard this hypothesis that cancer is a wound that never heals. So I decided to look at wound repair and compare to cancer. Now, the way I wound the skin was to selectively remove the skin epidermis and upmost dermis, challenging the remaining hair follicle stem cells to re-epithelialize the skin and restore the barrier. I followed the wounding response in the normal skin with co-staining of two lineage markers that I introduced to you, KL5 in red and SOX9 in green. As you can see in normal adult skin, these two markers delineate the epidermis and hair follicle respectively. And shortly after I made the wound, you can see that SOX9 you can see the SOX9 marked hair follicle stem cells are fine. But curiously, by day 3 to 5 post-wounding, these guys start to express KL5, resulting in co-expression with SOX9. But as the wounds heal, SOX9 and KL5 actually restore their homeostatic expression patterns, demarcating hair follicle and epidermis respectively. Therefore, lineage infidelity is a wound repair phenomenon, and which is hijacked by cancer. Now, the first question we set out to ask was, do the marker expression matter, right? So the answer is yes. When we deleted either SOX9 or KL5 in the squamous cancer, the tumors no longer are able to propagate in vivo. Now, what's also interesting is when I took a look at the residual tumors from the KL5 knockout samples, you'll see that the K5 stemness marker, which marks the proliferative basal cells, start to get depleted, whereas the K10 differentiation markers start to pick up. And that suggests that the KL5 is important to sustain tumor stemness, if you will, in these samples. Now, how about wound repair? In order to knock out my candidate genes in wound repair, what I did was to apply in vivo CRISPR by in utero injecting lentiviral construct carrying the CRE as well as the CRISPR guide into the CRE-inducible mice with a GFP reporter. Then I started gene ablation shortly after birth by tamoxifen injection, which now activates Cas9 as well as the GFP expression. As postnatal day 3, we split the skin, that is to enzymatically remove the epidermis and graft the dermis containing the green hair follicle onto a nude back, which is colorless. In a couple of weeks, we'll look at the GFP expression in a regenerated epidermis and simply ask who contributed to regeneration, was it the graft or was it the host? This is what we saw. So at P3, right before split, epidermis was similarly green across all conditions, suggesting equal labeling. And I hope you can see that KL5 is efficiently ablated, indicated by lack of the KL5 staining. Now at 2 weeks, grafted hair follicles from the wild-type control really contributed efficiently to the regenerative epidermis, reflected by high amount of GFP cells in the epidermis. In striking contrast, the KL5 knockout graft didn't contribute at all. The skin still heals, as you can see from the picture, but they were not green, and they are wild-type for KL5, suggesting they came actually from the host, not the graft. It is also obvious from the skin color that since the graft was from the back mice and the recipient is pink, you can easily tell who made the contribution, judging from the skin color. Now, without showing you the data, we also found that SOX9 is important also for this process. So is it just this 2 transcription factor, or what is more broader? So to this end, what we did was to purify stem cells from these 2 normal lineages, the hair follicle and epidermis, and also these stem cells, the hair follicle stem cell during wounding, along with SCCs. Now, we did RNA-seq and ATAC-seq to look at their transcriptional and epigenetic profiles, and this is where the striking surprise came. So as you can see here from the GC analysis, this is from RNA-seq, and you can safely conclude that if you find a gene that is increased in wounded stem cells compared to normal, it is very likely the same gene is also increasing in cancer stem cells. And same thing with downregulated gene, which I'm not showing here. This can also be seen from the PCA plot, which is the global RNA-seq profile. You can see that the epidermis and hair follicles are away from each other, where the cancer and wound stem cells here in pink and in orange here are coming together. Then I had this idea. Now, perhaps it is proliferation that dictates their similarity and then results in lineage infidelity, because wounded cells and cancer cells are both very proliferative. Now, I want to use a control. The proliferative cells I focused here were those hair follicle so-called transient amplified cells. Now, these are proliferating progenies of the hair follicle stem cells during a normal hair follicle regeneration. Surprisingly, these cells, despite going through the proliferation in vivo, they do not look like tumors at all. They also don't look like a wound. Note here that the scales are also very different, that there's absolutely no correlation whatsoever. So these results suggest that hair follicle undergo certain lineage plasticity in the wound and the cancer that are fundamentally different from those that are during normal hair follicle regeneration. When you think about this, it does make sense. During normal regeneration, hair follicles give rise to all lineages of the hair, right? But just the hair. And then upon wounding, however, their fates now become expanded. They regenerate not only hair, but also epidermis. And that's what's the plasticity comes in, right? So it makes sense to evoke a different mechanism and rewire the gene networks in order to achieve that plasticity. Now, we think that this is the exact state where the cancer is coming to hijacking. I wanted to give you another example here on genes that are absolutely not expressing in normal skin on the homeostasis, but hyperactivated in cancer and wound repair. And that is MIR21, the oncomere I mentioned earlier. Here, I'm showing you a region about 300 kb on mouse chromosome 11. You can see a dramatic induction of chromatin accessibility measured by ATAC-seq in these pink tracks, right? Compared to the FACS purified hair follicle and epidermal stem cells, which in green and red here in vivo. Now, these are our wounded cancer cells in pink. Another important point I wanted to make here, if you look at conditions where these chromatin regions become open, here is from the in vivo hair follicle stem cells when they put them in culture, which is, and also the hair, and also the epidermal stem cell in culture. Now, in fact, you can see that the culture cells look very much like the tumor cells, right? And then the wounded cells. In fact, we have the data to show, which I don't have time to show here, is that globally culture cells look like wounded or cancer cells rather than their in vivo counterparts. I want to emphasize this not because this is not because of bad culture conditions. These culture keratinocytes from skin tissue upon grafting into a mouse host, they can form skin. And our cultured stem cells of the hair follicle can grow hair on hairless mouse. So they are truly stem cells. Now, these examples really state the obvious, that is a cellular transcriptional and epigenetic response is directly determined by its microenvironment. And when stem cells are taken outside of their native niche and put into culture, they undergo a wound-like stress response. Now, back to cancer and the wound, I'm showing you this region because it harbors MIR21 that is completely silent in skin. And you can see from the in situ hybridization that they get hugely induced in the benign and malignant tumor in the skin. Now, it is important to notice that you are looking at 10 to 100 kb of putative active enhancer marked by H3K27 acetylation, not just plus minus 5 kb around the transcription start site of particular gene, right? In fact, many of the genes in the vicinity are not expressed, or their expression remain constantly remain constant irrespective of their stem cell conditions, right? You can also see that MIR21 is induced in the wound, but transiently in this in situ staining. So MIR21 is actually right here where the arrow is shows, and the primary transcript only occupies about 1 kb region. And you can see that its promoter region is actually open in all conditions, yet it is not expressed in the normal skin, and it has gained this huge enhancer around it in the cancer and in the wound, including in culture. What's interesting about MIR21 is besides tumor, it's also induced in many stress conditions, including inflammation such as psoriasis. And we could say that it is actually a poster child for stress response or a biomarker, and under certain contexts, it is functional target. Now, in fact, besides MIR21, we have many other genomic regions and genes showing such stress response. Here's another locus next to KL5, and we could test whether these peaks are truly stress-specific enhancer elements or they just random open chromatin, right? We can do that by enhancer bashing in vivo. We simply clone these Ps into a lentiviral reporter to drive GFP expression, and meanwhile, we have a ubiquitous promoter to drive H2B RP expression as internal control. We then introduce the lentiviral reporter into mouse skin by in utero infection. And then we let the mouse born. We simply ask, when and where do we see GFP activity? As expected, we found specific GFP activity only in the wounded and cancer skin stem cells, but not in the normal homeostatic skin. Remarkably, this also holds true with human SCC cells grafted into the mouse skin, and that includes both the skin SCC and the head neck SCC. Now, this is power, right? Because these regulatory elements are specific to stem cells undergoing lineage infidelity and stress response. One can imagine we can use them to design cancer-specific targeting strategies while sparing the normal tissues. And that also suggests that these enhancer elements are evolutionarily conserved. Now, next, what we wanted to ask here is what is driving this dramatic change, right, leading to lineage infidelity in wound and cancer. To answer this question, we went back to our open chromatin data and epigenetic regulation. Given the dramatic remodeling we've seen with MIR21 locus and other genomic regions, the fact that at open chromatin level, wound and tumor also closely resemble each other, right? They also diverge from the normal cells. This is ataxic data I'm showing here. So we wondered whether we could extract a global stress-specific signature. So long story short, we pinpointed here one of the ETS factor S2 to be highly enriched, having binding motifs on enhancers that are specific to cancer and wound. Showing here is such one of the peaks that I just showed you for the reported assays. Now, S2 protein is constitutively expressed in the skin, but it gets phosphorylated and activated by ERK downstream of RAS-MAP kinase signaling, which is a potent driver for SCC. So we set out to test whether S2 activation is sufficient to drive lineage infidelity. To do this, I used the in utero lenti injection technique that we like to do. We delivered into the TRE F2 cassette into a K14 RTTA mouse so that a doxycycline can induce this ETS2 expression. And it's a it's a phosphomimetic so that it will be constitutively active. Now, when we feed the mouse with doxy water, this is what you see. We found that SOX9 and KL5 now becomes co-expressed even in the normal skin. And I say it's normal, but it does have a hyperplasic response. It's not tumor yet. You do see that phospho-S2 and all these stress markers turning on. So this is prior to gross tumor development, and in itself is not sufficient to drive tumor. But you can see lineage infidelity actually occurs early. So it is not really a secondary effect from tumor formation. It is an early event. And the F2 is probably one of the key factors that is causal to this phenotype. So finally, why does cancer doesn't heal though, if it looks like the wound so much? So recalling the model that I told you about, hair follicle stem cell gets activated and send progenies to regenerate epidermis. During this process, SOX9 is turned off while KL5 is turned on, right? Now, what if we sustain KL5 and SOX9 double positive, right? Double expression by specifically artificially driving SOX9 expression. So I did that again with the in utero trick. This time I deliver a TRE SOX9 into a K14 RTTA embryo. And upon doxy, SOX9 will be constitutively active and cannot be turned off. And this is what we found. We found that when SOX9 is artificially sustained during wounding, you can see that the skin have hard time to heal, and all of them become a hyperplasic status. Again, this is not tumor yet. It's really just hyperplasia, but at least it suggests that SOX9 can make a wound that doesn't heal. So to summarize this part of the story, in the skin, we have two main lineages, hair follicle and epidermis. Each are maintained by its own resident stem cells expressing a set of lineage factors, and SOX9 for the hair follicle stem cells and KL5 for the epidermal stem cells. And hair follicle stem cells, in this case, only make hair under steady state. Now, upon wounding, stress transcription factors are induced. For example, ETS2 gets activated, which then lead to mixed lineage expression. And in the case of hair follicle stem cells, SOX9 cells become double positive for KL5 as well. and setting off lineage infidelity. Now, lineage infidelity signifies a functionally revived state that enables stem cells to expand their fates, but not only to make hair, but also regenerating epidermis to heal the wounds. And this is exactly the state that cancer hijacks. The difference being that the lineage infidelity is resolved when wounds heal, whereas it's sustained in cancer. Now, one of my lab's current interests is to characterize the molecular mechanisms driving cutaneous squamous cancers and related cancers, such as those in the head, neck, esophagus, and lung, and many other tissues. And we would like to understand the cancer driving pathways in these similar squamous cancers. What we find really fascinating is these strong convergence of squamous identities and pathways across many different tissues. And we hypothesize that such squamous lineage addiction could be exploited as potent cancer targets in these specific carcinoma patients. Now, we are very fortunate to be embedding ourselves here at MD Anderson with very strong clinical and translational groups across campus, and including everybody listed here across pathology, head, neck, and many others. So our goal is to really examine whether the cancer drivers we discovered using mouse genetics would be applicable to human squamous cancer, and if so, under what context and by what mechanisms. So for the last 10 or 15 minutes or so, what I'm going to do is switch gears now and tell you briefly the second story about aging. Now, this is my lab's work in collaboration with my previous mentor, Elaine Fuchs, and luckily the manuscript was gladly published during this COVID-19 time. So just like many other organs undergoing architectural alteration and functional decline during aging, the skin aging, and particularly the aging in hair follicle, is accompanied by a reduction in hair cycling and sometimes bald patches, as many of you know, leading to the notion that hair follicle stem cell might have been lost or undergone deteriorous decline during aging. Now, the mechanism, however, is not clear. And in particular, there's this interesting recent report that hair follicle stem cells undergo epidermal conversion during aging. I got really intrigued with this notion, recalling that stem cell lineage infidelity phenotype that I just talked to you about upon wounding. Now, hair follicle stem cells activate epidermal genes and eventually become epidermis to complete epidermal regeneration, right? This sounds very much like it's during aging, according to this paper, and I wondered perhaps lineage infidelity might also be in play during aging. So that was the start of we pursuing this story. In order to address these questions, we went ahead to fax purify various stem cells in the skin epithelia, including the bulge hair follicle stem cell from 2-month and 24-month aged mice, and subject them to single-cell RNA-seq. So our rationale is such that hair follicle stem cells, if indeed undergoing epidermal conversion, it is likely to be a gradual process. And if that's the case, we ought to be able to capture some transitional cellular states that are specific to the aged populations. Now, the surprise is that, however, this is not quite what we observed. Here's a quick glimpse of all the cell population visualized by T-SNE plot, combining the young and aged data. These are the expected cell lineages annotated using our known lineage-specific markers in the field. Now, for example, you can see bulge population here marked in the pink right here. Now, when we separated the young and aged samples, you can see it more clearly. It appeared that each population remain well defined in the aged skin epithelium, not just like in the young. They are almost identical, except for some cell number may be different in different populations. You see a clear-cut bulge cells in the aged hair follicle as well, the bottom line is we do not find a transitional population from bulge to epidermis in the aging sample. So our single-cell RNA data seems to suggest that at least it doesn't support a fate conversion model during aging. Now, in order to further test the possibility that hair follicle stem cell might have completely switched their fate in the bulge and lost, we examined a few selective lineage markers using direct immunostaining to compare young and aged. As you can see from these images, as we saw very well maintained hair follicle stem cells in the bulge, including K24 mark here and the SOX9 mark here. You can see the numbers still reduced in the aged, but they are there, right? And KL5, again, the epidermal marker marks epidermis. It doesn't go down to the bulge. They are not double positive for these markers. And K10, another epidermal marker, they stop right above the bulge, just in the age, just like young. So therefore, it appears that the age stem cells in the bulge is able somehow to maintain their lineage identity. It doesn't go to infidelity. So I wanted to show you again this global picture of RNA-seq, and you can see that in the light and dark green, you'll see the young and aged bulge cells, and then in the pink and red, you'll see the epidermis cells. They separate by their identity, not by their age, suggesting again the bulge aged bulge doesn't become epidermis, at least at the global level. I don't want to mislead you to think that there's not much change in aged skin or hair follicle, as in fact you can see from the picture that how dramatic the change is in the B6 wild type strain. That's also why we like to study skin. The phenotype is very obvious. I hope you can agree. So meanwhile, you can also see that interestingly on the left it's a young, right? On the right and in the middle are both age. They are wild type females, and both 24 months, and they housed on the exact the same conditions. But one you can see have a fairly good hair coat, the other is almost bald, right? So it's also interesting to think that aging in itself is a highly variable phenotype in human, just like in human as well. Now I'm going to show you a movie here, and hopefully the phenotype is clearly obvious to the audience. Now, what you see here in green is ECAT labeled bulges. And this is, ECAT is not specific to the bulge, but it serves a marker to enrich for the shape here for you. Now, this is a stereotypical two-bulge structure within the young hair follicle at a 2-month age. And since the mice had undergone 2 hair cycles, adding 1 bulge at a time, you see 2 bulges. The gray PCAT labels so-called hair germ, and this is a multipolar progenitor cells that fuel the hair growth. Now let's take a look at the mouse hair follicle at a 2-year age. I hope you can appreciate that rather than multiple bulges, reflective multiple hair cycles, you can only find 1 single bulge. And the mice at this age had undergone at least 7 to 8 hair cycles, right? This is from a hairy mouse. In a more dramatic case, this is from a balding region of the aged mice. The hair germ even seems to have collapsed. Besides, you only find 1 single bulge. And this is a remarkable phenotype. And we do think that, you can imagine over time, these hair follicles are no longer able to physically hold hairs, even though bulge in the hair follicle stem cells are still there. These structural changes eventually result in sparse hair coat and various degree of balding phenotype. Such degeneration of hair follicle structure might also be directly linked to hair follicle miniaturization phenotype in aged skin and alopecia diseases. So we are currently trying to understand what are the molecular mechanisms, understanding such dramatic architectural changes. And specifically, we are focusing on some extracellular matrix gene, including collagen function in this aging context. The bottom line is the stem cell themselves seems to be okay, and in terms of their lineage, right? So next, besides the stem cells, we thought to look beyond, right? We wanted to look at the whole tissue as a whole, and especially the microenvironment cell types, because we know that stem cells are intimately associated with their niche types and which regulate their function. And to this end, we focused on several cell types that have been associated with hair follicle stem cell activity. Now, not surprisingly, everything we look at during aging is changing. So just give you a few examples. Here are some sensory nerves known to wrap around hair follicles above the bulge region in the young, now becomes disoriented in the age. Here are some the erector pili muscles. These are the ones responsible for goosebumps and hair reactions. You can see them nicely attaching to the bulge hair follicle stem cells in the young, but become detached in the age. Finally, the T-rex cells are known to be associated with hair follicle regeneration and hair cycles. You can see, I hope you can appreciate here that they are strongly reduced in the aged skin. I'm not showing you the quantification here, but just to show you some pictures. You have abundant T-rex in the skin in the young, but it's reduced in the age. Now, the hard question is, with all these multifaceted factorial changes, how do we dissect their function? What is really causing the aging phenotype? So to answer this question, we thought to test the hair follicle activity in two concepts, and I have two last slides to share with you in terms of data. One scenario is to look at endogenous wound repair, where young hair follicle stem cells are interfacing with young niches, and aged hair follicle stem cells interfacing with aged niches. In the other scenario, we will purify the stem cells and mix match with young and aged microenvironment and see what happens. Now, in the endogenous wound repair, we went back to our dermal wounding technique where we selectively removed the epidermis and activated the bulge hair follicle stem cells to drive wound repair. As you can clearly see from the picture on the left, the yellow circled area, while the young has efficiently regenerated hair follicles also they appear black, the aged skin is defective in doing so, and they appear pink. The black color is a good indication of hair regeneration because melanocytes, which I didn't talk about at all, they actually undergo cyclic growth behavior together with hair follicles. They secrete melanin delivered to the hair follicles during the regeneration, suggesting that mature melanocyte differentiation is in synchrony with the complete hair follicle regeneration. It's not happening in the old, obviously. Now, as shown in the section, you can see that from microscopic level, you can see fully regenerating, growing hair follicles in the young, whereas quiescent looking, very small hair follicles in the aged. Now, I want to emphasize it's not because hair follicles never get activated in the age. I'm skipping a lot of data here, but we went back to look at earlier time point. We do find activated hair follicle markers, including sonic hedgehog and wing 10B as in situ hybridization, showing you here in both young and aged wounds. However, in the aged, specifically, the hair follicles somehow appear immature, and then they lack the black hair shafts, which again, you can see very clearly from the young skin, right? So this suggests that the aged hair follicle has impaired differentiation during wound repair compared to the young skin. Now, how about we switch their microenvironment? So this is what we did. We purified the hair follicle stem cells, and we mixed them with either young or aged environment and vice versa. And then we ask how do the hair regeneration appears. And we call it a chamber graft. It's a graft model onto a hairless nude mouse. So as expected, young hair follicle stem cells plus young dermis grow hair. Aged hair follicle cells plus aged dermis do not grow hair, right, in the corner. But what you can see here is, interestingly, you have robust hair growth when you graft aged hair follicle stem cells with young environment, which is the upper right corner here, and no hair growth when you graft young hair follicles with aged dermis, which is the left bottom corner here, picture. I apologize, I couldn't get my mouse working, but so here are the quantification also. So together, the punchline of this part is such that, first of all, aged hair follicle do not lose their lineage identity and are maintained in the aged skin. So there's hope, right? And second, strong environmental stimuli can override intrinsic deficit of aged hair follicle stem cells and can rejuvenate hair growth. So my lab now is setting out to dissect which cell types and signals are responsible hair follicle rejuvenation, and hopefully to make a broader impact on hair regeneration in aging and other related alopecia diseases. So with all that, I like to end my slides by thanking our funding agencies and give a loud shout out to my fantastic group members here, especially during this challenging time. That some of you, I know that, I don't know if you are here, but some of us have to stay home and put a hold on the career for the benefit of the big community. And some of you are actually stuck outside of the country and going through some really life-changing adversities. So you can see we like to go out and eat a lot before the pandemic. So I'm deeply grateful that the GLAB members are sticking together and trying our best to come out of this strong. I thank you for your attention, and I'll be happy to take any questions if there are any.
Beijing, wonderful presentation, a lot of very new and interesting data for all of us. And now let's start with a question. So first question, what's the role of nuclear hormones such as androgens on stem cell growth and differentiation?
Oh, I love the question. So it's going to be a very, I hear a lot of noise. Okay, better? Okay. So there are a lot of literature out there about nuclear hormone regulations of hair growth. And obviously, we all know that the male pattern baldness is intimately linked to heightened and deregulated androgen regulations, right? So there are many elegant mouse models to show that even overactivation or loss of nuclear hormone regulation could directly lead to baldness and hair growth defect. So we are interested actually in looking into certain aspect of these signaling pathways and hope to dissect. And the challenge here is really to understand different cell types and the regulation there. Which cell is sending the ligands, which cell is expressing the receptor? Is it epithelial? Is it in the stroma, the fibroblasts, or the immune cells even, right? And which signaling pathway can and also the redundancy. So this is a very interesting question, and I'm very general, didn't give specifics, but very, very interesting to pursue.
Very much. Another question. What's the real difference between cancer and normal wound healing in terms of SOX9 KLF5 expression? Cancer stem cells can express SOX9 KLF both, but wound healing is not. Is that right?
So I might not be very specific, but it depends on at which stage of wound healing we are looking at. So the immunostaining that I showed earlier, we actually see co-expression of SOX9 and KL5 in the hair follicle stem cells at the peak of wounding, during day 3 and day 5. But they don't stay always like that. The stem cells, the hair follicle stem cells actually lost SOX9 expression and switched to KL5 expression. That's why they become epidermis when the wounds heal. So that's we call the phase switch. But during the middle, they actually co-express both factors. And that's where you always see in the cancer, they always co-express these two factors.
Another question from my side. So did you check on the roles of non-coding DNA region in the genome? You show that very nice data on MIR21. Now the genome has this less known super enhancers, insulators, boundary regions. So these non-coding DNAs who probably very much affects the plasticity of a cell and so on.
Yeah, George, I really would love to collaborate with you on this particular question. There aren't large-scale study done in order to screen for non-coding regulator of, either wound repair or squamous cancer in the skin. There are a few really elegant small studies, single isolated studies, including Paul Cavari at California. So they have shown that some non-coding, long non-coding RNAs are important to regulate the stem cell, the epidermal stem cell differentiation, and presumably cancer as well. It's a little bit challenging. I think one of the limitations we have right now is the tools, because we do want to genetically testing the function of these non-coding regions or even the enhancers. So what we're hoping to do now, we have the CRISPR system setting up and go in the mouse, right? So we have the Cas9 mouse and we are screening for genes now. But I think that what we wanted to do is really to really adapt it to the non-coding regions, which has always been one of our major interests in the lab.
Nice, great direction of research. And one more question. Any association between MIR21, I think, expression and SOX9 KLF5 expression?
Yeah, correlation with the microRNA. Yes, but not directly. So MIR21 is induced during wounding, during in squamous cancer, and that's also where you find the SOX9 and KL5 co-expression. However, that said, I don't think that it holds true in, it's not a universally true phenomenon in other types of contexts. So I don't think that I shouldn't say I don't because I never examined. I don't have evidence that miR-21 directly regulates these transcription factor pathways. That's what we have. But they are sometimes correlated because, you know, they might share some common regulatory elements in their enhancers as well.
One question again. Will you comment on connections between lineage plasticity and mutations in cancer? Any idea about targeting plasticity in cancer?
Yeah, that's a good and difficult question. So this is one thing that we so far our methodology has limitations because we use a couple of gene models where we put clean genetic mutations into the mouse and we ask what is the consequence of the regulation, what is the plasticity. So the model we used was HRAS G12V driven squamous cancer. And sometimes we also put in TGF beta receptor knockout to make the tumor more malignant. We've tested in culture conditions some other genetic background like P53, but very few studies. So I cannot say that it holds universally true in other genetic mutation background, but we are very curious to actually examine. For example, in the skin, specifically as our colleague Curtis Pickering and Jeff Myers and others have shown that Notch is one of the top mutations in the cutaneous squam. And we are very curious to see whether such relationship actually holds true there. You know, the model that we tested, the skin model we tested was EGFR amplified tumor. And, you know, I will be really excited to see whether this plasticity really holds true across different genetic backgrounds.
And with this one, thank you again, Yejing. It was a wonderful presentation and a great lesson how you can look to normals to understand what's bad in cancer. And I want to thanks to all the participants. Remembering them to look on the web page for the presentation and saying that each day is passing, we are close to coming back at what we love more, to do research, fundamental, translational, clinical research. We are here, all the members of MD Anderson family. Doesn't matter how tough and rough the days are, we are here in order to do what we love the best, to enjoy science and to make discoveries. Thank you very much. And please keep in mind, Thursday and Friday we have other wonderful speakers, and I will make you also a new announcement because of you, because of the speakers, and because of the people who organized this webinar. It was a big success. We decided all together to continue during all summer on a format of once a week, Fridays 10 a.m. So starting in June, we will meet continuously, full summer long, 10 a.m. Fridays. And with this, thank you all. Be happy, enjoy science, and enjoy life. See you tomorrow, all of you, Thursday, all of you. Thank you.
Thank you, George.
Thank you, Jean. Wonderful, wonderful talk. Thank you again.